Hi, y'all. This is an extension of the scientific process or scientific method notes that we have already gone over. So, this is if you were going to set up an experiment on your own or you're going to create an experiment, how you would make, how we say, a good experiment. Criteria for a good experiment, we have an acronym for that called COMAR. It doesn't mean anything, the word doesn't mean anything, but it just COMAR stands for something, so it helps to remember what are some of the parts of a good experiment. So if you're going to set up an experiment, what is it going to look like? All right. So the C stands for control group. So do you have a group of individuals, living things, whatnot, that is um, does not have the independent variable added? Okay, because we can only test one variable at a time. Everything else must be constant. So if you're testing multiple variables and you don't have a control group, it's going to be a problem. Okay, so control group, step one, you need to have a group that does not have any variables added. Okay, but it should be a group of individuals or things that are similar to the other test subjects. Okay, one independent variable added. So O stands for one independent variable. So you don't want to have multiple variables. Like I said, everything else is constant, and you have one thing that you are testing, you are trying to figure out. M stands for many trials. If you just run the experiment once with no other test subjects, it's going to be difficult to prove that your experiment was successful or not based on your one trial. Okay, so we need to have many trials, and this might be like hundreds of trials. Okay, depending on the situation or the experiment, it could be a handful of trials versus hundreds of trials. So it depends on, like I said, the experiment. A stands for accurate and scientific measurements used. So, what are the tools that you're using to do the experiment, okay, and to collect the data, the dependent variable that you are trying to figure out. And R stands for relevant data, okay? So, the right side box here, just some pro tips: test only one variable at a time. Everything else must be constant. We call those controlled variables. You need to have a control group. So, if it's like a medication trial to see if a medication works, that control group would be given a placebo. Okay, for example, that's like a pill that does not have the active ingredients. It's just sugar pill. Okay, um, a conclusion should answer that question you asked. So once you've done the experiment, you've collected data, analyzed, and drawn your conclusion. Does it answer the question you originally asked that led to your hypothesis? Yes or no? Okay. The experiment should be able to be reproduced. So it's not something that's so crazy, so expensive, so out of this world that nobody can ever, replicate the experiment. You need to have something that can be replicated, or else your experiment is probably not going to be very credible. There need to be multiple tests, so many trials. Each test is given enough time to be measured properly. Okay, some tests take a while. So if you're dealing with plants, for example, that might take a couple months for the plant to grow or whatnot. Proper measurements must be used. So those are things that need to be measurable. So measurements need to be measurable. So they need to be using tools such as rulers, scales, balances, etc. You're collecting quantitative data. There should be a control group always. The conclusion is based on measurable data. Okay, so you're not testing like how much fun something is, because fun is not something that's measurable. Okay, and ethical and moral questions are not answered by science. That's a big one. Okay, so we're gonna get those ethical and moral questions out of here. We don't want to worry about those. We want to be doing experiments that can be replicated, duplicated, measured, whatnot. Okay, so how do you solve these COMAR problems? So I'm going to give you an experiment, and you're going to try to figure out what's wrong with the experiment. Okay, that's what you're trying to do. You're trying to pick apart the experiment as if you were a grader. You're grading the experiment. So what I suggest you do is you write COMAR at the top of the page. You search for each component, cross out any of the ones that you find, and at the end, if any of them remain, you need to double check and make sure if anything's missing. All right, so let's practice. The students in the room 249 were concerned about how cold the room was. They decided to measure the temperature every day in 5 different locations using a thermometer. Then they averaged all of the temperatures. After a month, they determined that the room was too cold. So, using our Colmar, okay, it looks like we are in one room only. Okay, so all students are experiencing the temperature. They're experiencing how cold it is. They don't really have anything to compare to. So if they're saying it's cold, but like how cold? Like what are we comparing it to? So we don't really have a control group here. Okay. So let's move on. They decided to measure the temperature every day in 5 different locations using a thermometer. Okay. So it looks like they're using a thermometer, so that's good. And they are measuring the temperature every day, so that's constant. And it's in 5 different locations around the room, so it's not just one cold spot. Okay, so it looks good so far. Looks like we've got one independent variable. We got many trials. I think we're good here. Issue for the accurate and scientific measurements. Yes, we're using a thermometer, but they averaged all the temperatures. Okay. Yes, I understand the averaging of the 5 temperatures of the different locations in the room every day. I think that would be a good idea. But we should see the difference between day to day. See if there is a difference based on maybe how cold it is outside, or maybe the building's just hot. Okay, so that A for Colmar seems a little odd. And then they averaged all the temperatures. After a month, they determined that the room was too cold. So that at the end there, that is not very relevant, okay? Because like how cold? They not once did we see an actual temperature in degrees Celsius or degrees Fahrenheit. Okay, so here was my analysis of that. Okay, so we're gonna scroll through here. Okay, so this one's good. Students decided to experiment how much water and fertilizer will grow the tallest tomato plants. They were given one set of plants, which are about 50 plants. The recommended values of water every other day and no fertilizer. Then they gave another set of plants water every day and fertilizer every day. At the end of the experiment, they take the mass of the tomatoes. So what I want you to do right now is I want you to pause and I want you to write Colmar on your paper, on your iPad, wherever you're watching this, and see if you can pick apart this experiment. Okay, and talk to somebody about what you think, what your results are on this, on your findings for Colmar. Thanks for watching. Bye.